Thank you very much for the invitation. 2 years ago, we had a small endemic of dengue fever in Tokyo. And we don't have Aedes aegypti in my country, so Aedes albopictus is the major vector of dengue. And at this moment, Aedes albopictus in Japan is totally susceptible to all of insecticides. But we are worrying about developing insecticide resistance in near future. So I'm collecting insecticide-resistant mosquitoes from the world and studying molecular mechanisms of pyrethroid resistance. Today's my topic is genetics of insecticide resistance, especially about target site modifications. But there are at least 29 mode of actions for insecticides, so it is impossible for me to overview all of them. So today I would like to focus on only pyrethroid resistance of Aedes mosquitoes. These two mosquitoes are the major vector of arbovirus, including Zika, dengue, and chikungunya. But there are some differences in behavior in these two species. For example, in Aedes aegypti, like to stay inside of houses, and they like to feed on human. But Aedes albopictus usually, sometimes they come into houses, but they usually stay outside of houses, and they feed from variety of blood sources. So Aedes aegypti can easily be selected by insecticide, but Aedes albopictus show relatively lower level of resistance. I plotted resistance ratio so far reported in the world. Each plot shows each resistance ratio, each population of resistance ratio. You can see that Aedes aegypti developed higher level of resistance to pyrethroid than Aedes albopictus. Sometimes Aedes aegypti showed more than 600-fold of resistance. But Aedes albopictus showed less than 10-fold of resistance. The target site of pyrethroid is sodium channel, VSS, voltage-sensitive sodium channel. And so far, several amino acid substitutions have been reported from Aedes mosquitoes. But not all of them confer resistance. So some researchers are characterizing each amino acid substitutions by neurophysiological studies. And at this point, we know at least three these mutations are involved in the resistance to pyrethroid. And this is a list of sodium channel mutations, so-called KDR, reported from Aedes aegypti. Amino acid F1534C has been reported from both Asian and Latin American countries. But valine 1016G has been reported from only Asian countries. On the other hand, in Aedes albopictus, only two amino acid substitutions, F1534C or S, have been reported. This mutation, we detected this F1534C mutations for the first time from the Singapore strain, Singapore population, by the collaboration study with Singapore NEA. And this year, Chinese group reported same amino acid substitutions from China and Greece. Also, F1534S was reported. By doing neurophysiological studies, we tried to compare the susceptibility of these 5 sodium channels to permethrin and delta methrin. And we found several interesting phenomena by doing this. First of all, we found that VG type sodium channel is brings more higher resistance than FC type mutations. VG type mutations brings at least 4 times of resistance than F1534C. So VG type mutation is more important than FC type mutations. And so far, VG type mutation has not been detected from Aedes albopictus. But Aedes albopictus is also gradually developing resistance to pyrethroids recently. So I suppose that in near future, this VG type mutation is also detected from Aedes albopictus. And we also found that this FC type mutation shows lower susceptibility to permethrin, but not to delta methrin. This finding actually has been reported by Dr. Cardon's research group using sodium channel of German cockroaches. They found that FC type mutation confer type 1 pyrethroid, but not to type 2 pyrethroid. There are some arguments about type of pyrethroids, type 1 and type 2. But technically, pyrethroids having this alpha cyano group can be called type 2 pyrethroid. So as I mentioned before, in Aedes albopictus, only FC type sodium mutations have been found. So for such resistant mosquitoes, type 2 pyrethroids are still effective, highly effective. And we don't know about this newly found FS type mutation. So we need to characterize this type of mutation in Aedes albopictus. We also found that S989P mutation also conferred to delta methrin resistance, but not to permethrin. And finally, I would like to emphasize this important finding. We also generated a sodium channel gene having all of these 3 mutations and injected to genopath oocyte and checked the susceptibility of sodium channel. And we found that such sodium channel having triple mutations showed extremely high level of resistance compared to other single or double mutations. Such triple mutations can be generated very easily by only one crossing over event in the mosquito. But unfortunately, such mosquitoes having triple mutations have been recently confirmed from at least 3 countries, including Myanmar, Indonesia, and Singapore. So I think this is a serious problem. We need to investigate the fitness cost of such mosquitoes. So here I would like to summarize the mission of WIN that I think to overcome KDR resistance. First of all, we need to understand the types and frequency of the KDR genes of Aedes mosquitoes comprehensively in the world. And we need to develop a database of KDR genes worldwide in detail. And secondly, we need to understand cross-resistance level of each KDR mosquito. We need to know structure resistance level relationship. I mean, not one KDR gene does not make resistance to all of the pyrethroids. Some pyrethroids may be effective. Some pyrethroids does not effective. So we need to know structure resistance level relationship. By doing such experiment, we may be able to design new pyrethroid that is highly effective to KDR mosquitoes. I think we can modify pyrethroids that overcome resistance. Here is one example. Recently, our research group published a paper about housefly, KDR, and super KDR, KDR and super KDR houseflies. Generally, super KDR houseflies shows higher level of resistance than KDR flies, like this. But among many pyrethroids, we found that super KDR is less resistance than KDR to some pyrethroids, including fenfluthrin, tefluthrin, transfluthrin. So I think this gives us a hint to design new insecticides that overcome KDR type mosquitoes. That is why I'm collecting many types of KDR mosquitoes from the world and doing tremendous amount of bioassays now. And this is the last slide. We also are we are preparing to construct database of KDR gene of the world. This is example of a dengue virus serotype, like this. But I think we need to make such database for KDR genes as well. And that will help maybe good management of resistant mosquitoes in the future. Thank you very much.